listen or see about this new way of doing genomics, bioinformatics, and biology in general. I've entitled this presentation Agentic Genomics because, in a way, it's a reflection of the work that I've been doing now for a couple of years since I began leading University of Westminster MSc degree on AI and digital health, and also through my fellowship with the Alan Turing Institute. All of this together with my previous bioinformatics background, in which I've been working for about 20 years now, and being in London, all of these ingredients have produced a cocktail, which I think hopefully is going to be a little bit different and certainly I hope innovative, at least from your own perspective. And I look forward to your questions, comments, and anything in between. So I'm going to be talking about agentic genomics and living through the AI revolution as a genomicist. And as I said, this is going to be a little bit, not a little bit, but actually the whole of it is going to be about my vision in terms of the state of the art between the intersection of the AI revolution, genomics, and biology in general, and how this is going to change. Hopefully, I'll give you a little bit of a vista of what the future of being a biology would be in a few years' time. So let me just, I guess I have no, I have no evidence to support this claim. So for what it's worth, I would argue, and I'm happy to be contradicted, that genomics is probably the fastest biology subdomain being transformed by AI. And the reason I think that is twofold. First, because genomics is really highly computational, and you can't really do any type of genome analysis without a massive access to massive data sets and massive compute. And guess what? Data sets and compute are the perfect ground for AI to flourish if you're able to embrace all of these amazing new frontier models that are emerging as we speak. So I'm going to contend that genomics is the fastest biology subdomain in terms of AI adoption. And as I said, it has huge amount of data, which is good for AI. It has huge amount of necessary compute. And so hopefully this will help us be the sort of illustration of the peak of the iceberg as to what precision medicine, the medicine of the future, could look like. So what do I mean by agentic AI? If you are in the sort of AI circles, you will have heard about agentic AI, and you will have heard it again and again and again and again to the point that it's kind of a buzzword. If you are not in the AI crowd, you will not have heard of it. So I'm going to assume that perhaps it might be worth to define what I mean by agentic AI. So agentic AI is really what 2026 is about. So first we started with the ChatGPT revolution in 2023, end of 2023. Then we moved into the big, hit of DeepSeek, which was this Chinese model, which was able to have comparable intelligence to ChatGPT at a much cheaper price. And then if I am really talking from the heart, I believe that in the past 2-3 months, there has been an inflection point in terms of what's possible. And I have had the luck to be in the appropriate circles to be able to appreciate what's happened. And really, some people, and I would actually say that without trying to be too sort of how can I say theatrical about it, that we've reached a point of inflection in which perhaps we could be now in a situation where we have artificial general intelligence. Okay. And by that, I don't mean a chat box that gives you relevant answers. I mean robots, and by robots I mean cron, programs a little bit like Alexas. You have heard of Amazon Alexa. It's constantly running all the time, and it's able to listen to you anytime, and you can give their orders, and then she might be able to answer, and she might be able to do things in the background to give you the appropriate response. So in other words, something like that has happened at a level which is being democratized by some of the frontier AI labs, whereby AI systems can now autonomously install tools, deploy code, and run analysis all on their own without having any human input in it. So in other words, they're displaying agency. And not just that, they can use the power of the large language models intelligence to be able to really reason, find solutions, try specific hypotheses, realizing that they don't work, coming back and going through another decision tree branch where they can really act almost as if they were a human employee, right? So that's really what I mean by agentic AI, is this capacity whereby AI-directed robots, if you will, can now be accessible, take decisions on their own, and basically write, read, and do a lot of the tasks digitally that someone would be able to do in the computer. Right? So as long as what you do is sort of reducible to the computer and can be done in the computer, in principle, all of that could be potentially automated and then acquired as skills that can be called, that can be executed for a specific function, and you communicate with the robot in plain English, as you would talk to someone as if you were face to face, right? So I think that, to me, with a number of caveats, because if when you get into the nitty-gritty of it, it doesn't always work. It may hallucinate, but you could say I could say that 50% of the time, every time I prompt any of my agents, which, by the way, I can talk to my agents via my phone. Yeah, so not just the chat box, and I'm actually going to demonstrate that in a minute. They are able to make decisions and get back to you with answers through a variety of intelligent decisions based on the context that they have for that particular prompt. Okay, so let me show you how this actually works. Okay, so I'm going to see if I can, so you can see me, right? I'm going to demonstrate live my robot. So let me introduce you to my robot. Her name is Roboteri, and I'm going to share it so you can see it. So this is Roboteri. hopefully all of you have heard of a messaging app called Telegram, which is, is very similar to WhatsApp, okay? So Roboteri is a 24 hours, seven days a week awake chatbot. I can talk to anytime I want to from my Telegram application. So I'm going to use it from the computer. So here I've got my Telegram. You can see my robot. Yeah, so this is Roboteri. Yeah, and as I said, it's currently working anytime. I can type and say to hey, Roboteri, how are you? Yeah, so it's, how are you? I'm a collection of algorithms, but I'm ready to help you. What can I do for you today? Okay. So, hey, this is what she answers. Now, I'm going to do something a little bit more exciting than just a random message, okay? I want you to imagine, yeah, that you've been prescribed a drug and you have your own personal genome. That is all of the data corresponding to your own genetics that has been sequenced. And imagine that you've uploaded that into Roboteri's memory. And I'm going to go to a pharmacy to get the drug I've been prescribed. So I'm going to share my whole screen so you can see the whole thing as it is. Okay. So I have my presentation here. Yeah, you can see. Can you all see that? Sorry, I'm not able to see the chat right now, so I'm not sure if you are talking to me or not. So I'm going to make this bigger. You can see in here that, I mean, I have that picture, yeah? Or maybe I could just go and show you another picture that I have here. Just a moment, so you'll see. I've got my skills here in my apps. So I've got that demo, my skills. Where are the skills? Here are the skills. So I've got a number of demo images, okay, which I'm going to demonstrate here. So you basically can see, so this is, I'm in the pharmacy, yeah, and my robot has information about the genome. And there's something called, that you might have heard, pharmacogenomics. So pharmacogenomics is the science that allows you to predict your ability to metabolize a particular drug based on your genetic data. Okay, so this is incredibly useful because you can predict the likely dosage that works best for you whenever you are being prescribed a drug. And there's a lot of evidence and support to be able to recognize that. Okay, so now if you could see, so I'm going to have this picture. You imagine that I am in the pharmacy or the chemist, and here is Roboteri. You're gonna see that live demonstrated. So look at this window, Roboteri, where it is. I'm going to talk to Roboteri via my phone. And basically what I'm going to do now is that I'm going to go to Roboteri, and I'm going to look at this corner, okay? I'm going to take a picture of Roboteri, a picture of this drug box, yeah, and I'm going to send it to Roboteri. You will see it in the corner of my screen. Yeah, so I've now sent that picture. Can you see? That's the picture. And what's going to happen behind the scenes is that Roboteri is going to process that image, is going to interpret the drug underneath it, is going to check it against FDA recommendations for what the appropriate dosage is, and it's going to compare those recommendations against my own personal genome. And then it's gonna, based on my genome, it's gonna give me a recommendation. And okay, as I said, it's my own personal genome. And this is real genetic data, okay? And guess what? It predicts recommendation, avoid, not to use. This is warfarin, which is actually the most prescribed anticoagulant, i.e. blood thinner, in the world. And I didn't know this before I actually, for some random choice, I decided to test that drug using my genome. And I've learned that if I am given the normal dosage of warfarin, you can see here in the report that I can bleed to death, basically. Okay, and this thing is constantly running in the background. I can talk to it. I've got it on my mobile phone. It has the intelligence to be able to interpret and give me tremendously useful. You could imagine, right? If you code your genome and you are being prescribed a drug, you have, I mean, there are so many drugs. How can you keep track of all of the, how can you keep track of all of the potential drugs and how they would relate to your genome? So, I mean, hopefully this makes a little bit of sense in terms of the amazing stuff that we can now do using artificial intelligence. So, as I said, this gives us a power of reasoning that would not have been possible even a few months ago. And this is happening at a rate that is accelerating, as you have probably noticed, right? The models that you were using 6 months ago, or ChatGPT, or any of the other Gemini, you name it, any of the other models, they are 10 times, 20 times, 100 times better than the ones you were today, than the ones you were using 6 months ago. So we are in a new wild west where the possibilities and of course the implications as well in terms of safety, in terms of who is going to control this data and so on, are incredibly new. So hopefully this live demo has given you a little bit to think about. And so we're going to go back to the slides. Hopefully you guys have seen this, no problem. I'm going to share back my slides. I see that you haven't written in the chat yet, but welcome to write in the chat or ask if you are interested to ask any questions. Okay, so I'm going to go back to my presentation. So here is my presentation, and hopefully you can see that from a photo we can identify a drug, match the brand and generic name to a specific drug, then read real genotype data, look at pharmacogenomics recommendations, and provide a personalized dosage card generated. All of that from your mobile phone without having to really, you can take it anywhere, right? So this is just a very simple but hopefully poignant type of example that you can see in terms of agentic AI, right? So where and this is obviously an agent. You see my robot, which is an agent. So, and it's an agent because it's always waiting, it's always on, and it's able to make decisions and intelligently think and operate a chain of thought, as we call it, and provide relevant results back built on a powerful intelligence behind, coming from a large language model such as ChatGPT and so on, which is what was providing the layer of intelligence in the background. So let's carry on. So I have two robots, actually. One of them is Robotary, which I've demonstrated. Currently, it's able to understand 15 research commands, and it has automation and things underneath. I also have another one called RoboIsaac. That one I'm keeping private because it's more, I use it for my own personal purposes. But again, it's 24/7 running all the time, and it helps me with idea development, strategic thinking, and decision support. And it helps me in a relevant way because I fed it with around 50,000 documents, which are relevant to me, Apple Notes and things like that. So it has a very good understanding of about 10 years of context in terms of how I think, what my priorities are, and so on. And again, this is just the tip of the iceberg in terms of what's happening. So hopefully this has given you a window into the future in terms of what agentic genomics is going to look like. And again, I'm not claiming that this is going to be necessarily what's gonna stick, but I think this is a very, very new type of application where I'm able to demonstrate something which is incredibly useful, as you could imagine, in terms of your own health. So this new way of thinking accelerate the generation of code and deployment, whereby now people like me, I mean, I have coded quite a bit, but I wouldn't consider myself a top professional software engineer. But now I don't have to worry so much about the technical implementation because the current intelligence will allow me to develop this kind of applications without even writing a line of code. Yeah. So anything to do with tasks which are heavily computational and becoming automated are going to really being able to help you if you're able to harness them in the appropriate way. Okay, it will help you with literature review and synthesis, figure generation and data wrangling, code translation across languages, and as well as web app deployment in hours. That said, this kind of intelligence is not going to substitute for deep thinking. And this is one of the main messages I want to transmit in this lecture. I mean, obviously the sort of easy jobs, you can make the decision. I've heard the comment like, you know, oh, this artificial intelligence is gonna make us dumb. Well, maybe if you choose to, but if you use it for your own personal enlightenment, as I say, for your own super enlightenment, then it's going to really push you up like a rocket. Yeah, in terms of your capacity to produce, but the bottleneck is not going to be the hypothesis generation or the judgment. I think now what's really important, what's really in demand is the judgment. Okay, so anything, especially anything that could potentially have strong implications in terms of getting you into trouble, that, you know, like sending an email, it can write emails for you, it can develop applications for you, but at the end of the day, if you keep the human outside of the loop, things can go terribly wrong. So my rule of thumb when using AI, you know, if the stakes are very high, then you need to have a human there in the loop, because artificial intelligence is not going to do the discovery for you. At least the one that I can currently experience is able to accelerate discovery. It's able to automate the production of and deployment of the required analysis, but it's not going to substitute for your deep expertise. And I don't agree with those who think that, you know, you shouldn't learn how to program, you shouldn't really study because everything is going to be based on the AI thinking. And I think that's the wrong approach, because at the end of the day, where the real contributions are going to happen is not going to be in the stuff that AI can give you. It's going to be in the edge cases. So the sort of thinking outside of the box perspective that wouldn't be captured by the mainstream thinking. And so if you are someone who is used to think outside of the box, you're going to have a huge amount of advantage in this new world that we are experiencing. So this is summarized with this message here, that deep intelligence really is the bottleneck. It's not the processing power. And the final sort of human experience of all of the different possible exceptions that we in our own personal experience live, that can be substituted by a 1 million token window, which basically starts afresh every time you create a new session, right? And so there are this is not necessarily a limitation, actually, it's a feature. So and I think that's from my perspective is the right approach, you know, how to harness that context window that you're going to have in your model, so that you can then direct it to the next level in terms of intelligence production. And you have to think about the fact that it will never be able to substitute human intelligence. It will substitute maybe 80% of the cases, and that will be for those people who decide not to go deeper and not to really explore the edge cases, as I said. You have to think about artificial intelligence and specifically applied to genomics, but also to any other field, as alien intelligence, which is powerful, but it's not going to think exactly the same. And again, it's not going to have the nuance that human experience can give. And so rather than substituting it, what you do is you drive it, you know, like a conductor in an orchestra. You know, you tell the musicians when they need to play, at what intensity, and so you become a coordinator rather than, and at the end of the day, the final verifiable filter before you can share the stuff that you want to produce, in this case genomics-based discovery. So it is your responsibility that whatever you produce, then you are able to verify it, because the lazy way will be caught very easily. I'm going to go into the examples of where that can be caught. I mean, the fact that also we have the current models trained with data that already exists. Well, as it happens, most of the data from which precision medicine is actually being trained for generation of these intelligent models is based on patients of European ancestry, Northern European ancestry. So in other words, we have the situation where all of those models have been incredibly well trained with a small percentage of the global population. And the further you are genetically from this population from which those models have been trained, the less accurate the models are going to be, the more they're going to hallucinate, and the less context they're going to have to build their specific predictions. I'm going to go into that into detail now. So let me give you an example. I'm going to check if there's any questions there. Okay, no questions yet. So here I'm basically displaying the countries that currently are part of the International Health Consortium of biobanks. So we have a lot of data from a few countries, such as the UK, and most countries go unrepresented, which means that the information related to populations within these countries is incredibly sparse. And even if we measure the amount of information that we have within the countries for which we have global data repositories, health data repositories for research, you're going to find that none of them really have a good coverage of all of the World Health Organization priorities as described by the global burden of disease, which is probably one of the most accurate analysis per country, per disease, per population, statistics currently available for anyone to use and download. So this is part of my own research. And looking at the data banks, which are currently one of the main biomedical data banks, which are one of the main drivers for production of research, the vast majority of the data that exists is actually incredibly misaligned with World Health Organizations. What do I mean by misalignment? So there's the World Health Organization developed a measurement called DALY. So DALY is DALY. DALY means disability adjusted life years. Okay, and it's basically a standard measurement, normalized, that can be used to measure the amount of disease burden as related to any given disease. So there are diseases such as, for instance, stroke, that have millions of DALYs. Yeah, so there's a huge amount of burden of disease related to stroke, or diabetes type 2, or cardiovascular diseases. So there's a huge amount of DALYs. And there are five diseases: diabetes type 2, inflammatory bowel disease, coronary artery disease, breast cancer, and prostate cancer. These 5 diseases, 20% of all deaths in industrialized nations are implicated with any of these 5 diseases. Okay, so and this is the diseases that people in the industrialized nations die. But of course, we have 1.5 billion people in the global south that live or that die of things like malaria, schistosomiasis, gout. So things which are not really what people die in the Western industrialized nations. And the point I'm trying to make here is that the vast majority of the current precision medicine understanding works really well when you use artificial intelligence if it's related to diseases of the global north. If you use or query ChatGPT or Gemini or Claude or Perplexity, if you query any of these diseases, any of these engines or frontier models by any of the diseases that affect the global south, like the ones I've just mentioned, we can predictably guess how much bias they're going to be. And we've been able to measure that, particularly for those diseases where that have a huge amount of DALYs, but very, very little research output and knowledge available in the research literature, the global research literature, places like PubMed, you find that the algorithms, the LLMs, the Geminis of the world perform a lot worse whenever you ask them a question about those diseases than if you ask them a question on type 2 diabetes and so on. And we can reliably predict the amount of hallucination based on some of the research I have conducted. So one of the things that we are trying to do as a consequence is to be able to measure so that we can ensure, even if we're not able to produce the data, because the data production for health data sets is incredibly expensive, is sensitive, and you have to have a very strong infrastructure. But at least from my perspective, one of the ways which we can ameliorate the existing health inequities is by being able to measure precisely how disconnected any of those corpuses of research or outputs are to the mainstream amount of research literature, biomedical research literature. And this is something that we find. I'm not going to go into the details, but what I'm trying to say here is that there's a negative spiral here, which we are trying to sort of tell the world. So the fact that you don't have the infrastructure, nor the money, nor the ability to produce the health data for specific diseases which are not priorities for the global north, for the rich nations, that means you have less knowledge. That less knowledge means that current diseases advancements actually fit less into or is less applicable to that knowledge which is isolated. It means that you have fewer clinical trials for those diseases, and it means that there is less research, and it means there is more inaccuracies in the artificial intelligence. And as a consequence, clinical decisions are worse, there are treatment gaps, and so on. And the funny thing is that, again, one of the things that I keep saying is that, you know, all of the diagnoses, all of the specific current guidelines that we have for diagnosis, treating patients, they all have been tailored or researched on a very small set of populations, which tends to be white, wealthy, healthy, Northern European ancestry. So there is this amplification cascade whereby the lack of data, or what I call data invisibility, feeds into the epistemic invisibility, i.e. the knowledge that exists for those diseases, which feeds into the structural invisibility, because that means that we know less, so it's less attractive to invest, so we have less infrastructure. And because we have less infrastructure, then we go back to, we're going to have less data. Because we have less data, we have less knowledge. Because we have less knowledge, then we don't invest. So there is this compounding negative spiral, or vicious spiral, that we've been able to measure very reliably. And here you can see one of the examples, okay? We've developed something called the semantic isolation index. So by semantic isolation is this idea that if we're able to represent, like we can now, we can represent all of the research publications that have been related to a particular disease. We can represent it in the same type of models or vectors as large language models do. You know, I'm not going to go into details, but this particular representation shows the semantic similarity. So you have these diseases each disease is a dot, and I've colored them according to, are they infectious disease, injury, mental, neglected, cardiovascular disease, cancer, neglected tropical diseases. And so you can see also the size has to do with the size of the circle, has to do with the amount of published literature that exists. By the way, I generated this graph utilizing 13.1 million papers from PubMed. Okay, so I downloaded 13.1 million papers from PubMed that relate to the 175 global burden of disease classification that comes from the World Health Organization. I plot them in terms of distance between each other. So in other words, I'm basically reducing all of the body of knowledge reducing them into two dimensions, whereby circles that are closer to each other mean that they are more semantically related to each other. Yeah. So I have a huge matrix of similarity. I think it has like 750 dimensions. Each of these dimensions represent a specific variant of the body of knowledge that has been published for each of those diseases. I have reduced those 750 dimensions of measurement of semantic similarity into two dimensions, which is what you see here. And basically the point of this graph is that the diseases that tend to be most isolated are precisely the ones that correspond to neglected tropical diseases and diseases from the global south. Yeah. And so here we have a very, very clear way to see that there is a lot of there is a negative correlation between the amount of body of literature that exists for any given disease and how distant that literature in terms of semantics is from the main body of knowledge, which, as you can see, is well clustered on this side. And you may ask, so why is this important? Why should I care about this semantic similarity index? Well, because let's think of a particular disease. Let's think of COVID, okay? So before we had the pandemic, COVID was a tremendously, coronavirus research was completely neglected. Yeah, so I have the data, I'm not showing it here, but it was incredibly isolated semantically. But now, after the pandemic, there were huge resources that were given for COVID. And now if we look at COVID, it's a lot closer to the body of knowledge, okay? And what that means is that if you are closer to the main body of knowledge, which means the main discourse in terms of the technologies, in terms of the advancements that are happening in immunology, in cell biology, in all the different technologies, it means that those diseases that are closest, they are more likely to benefit from those advancements. Whereas if you are far away, the sort of language is going to remain very isolated. It's going to be disconnected, and we can basically trace that as a lack of development in terms of scientific advances. So it's a lot more difficult for these diseases to actually benefit from the main advancements that are happening where most of the research is happening. Okay, I know this is can be potentially abstract, but this is one of the research points that we are currently researching as is in the process of being published. So now let's move on. So again, here we are plotting the semantic isolation index for neglected tropical diseases. things like Ebola, like Zika virus, and things like that. And the semantic isolation index for all of the other diseases. And what you can see is that and this is a very sort of abstract way of measuring distance from the main body of knowledge. And you can see that neglected tropical diseases are further or are higher in terms of distance from where the main body of knowledge is. We can see that in another visualization here. You can see, sorry, they're all here. Schistosomiasis, trachoma, they're all here represented, and they are far from those where the main body of research is happening. And the highest distance diseases from the main body of knowledge, as seen here in purple, they tend to be precisely with neglected tropical diseases. So diseases that affect 1.5 billion of the global population, but they receive hardly any attention in our understanding of and in the development of resources and diseases infrastructure. What that means is that, again, this point where there is a compound interest. So here we have an example of breast cancer. So the breast cancer characterization in European populations is 30 times better. The genetic models that are different from Europeans, the further you are, some of them, like Africans, lose 80% of the accuracy in genetic models for prediction of risk. And from what I showed here, neglected tropical diseases have a 44% greater amount on average of isolation, semantic isolation, with respect to the other diseases. So if we think back into pharmacogenomics, there is this gene called CYP2D6, which currently around 25% of all prescribed drugs are metabolized for this particular gene. So we have guidelines that exist for dosing based on this particular genetic gene and its variation. However, the guidelines rarely are implemented equitably across different populations from the world, which means that if you use the current recommendations and you are not from the represented population from which this data was generated, there's a higher likelihood for you to experience adverse drug reactions. So some of the other type of work that I've been developing also include genetic data coming from by countries from the global south. In this case, I've had a lot of work with the Peruvian genome projects. And if you don't mind, I'm going to have a pause for a break, a bio break for 2 minutes, and I'll be back if that's okay. So I'm gonna pause, and then we carry on into 2 minutes. I'm not gonna change anything. I'm just gonna go be away for 2 minutes and come back. See you in a moment. So welcome back, everybody. Let's now start the second part of this lecture. We're going to change gears a little bit, and I'm going to be talking about some of the work that myself and my collaborators have been doing in precisely in a country from the global south, which is Peru. Peru is an incredibly diverse country that, to my surprise, is in terms of the human, the number of indigenous populations, as well as the number of people who are of indigenous descent, is the biggest in the whole of Latin America, ahead of Brazil, which to me was surprising. And the reason for this is because Peru has an incredibly heterogeneous geography. It has the coast, which is completely desert, and it goes from north to south, and then it's crossed by the Andean chain of mountains, which gets up to 6,000 meters high, and there's a lot of mountain which acts as a barrier, and there's a huge amount of different populations that have been throughout millennia living in that very, very specific geographical characteristics. High altitude, quite dry, and also a huge amount of ultraviolet exposure. Then you have places like Titicaca Lake, which is a massive ecosystem, and it's in its own right, 4,000 meters high. So there's an incredible amount of diversity, and I'm not mentioning the Amazon, which is actually of all these three regions, coast, mountain, and Amazon. Amazon is the largest. So Peru is the country after Brazil that has the greatest amount of Amazon, like jungle, proper jungle land after Brazil. So it's incredibly diverse and wonderful place to go and visit. And I've now been working with a number of researchers for a number of years on understanding precisely the genetic adaptations that through evolution to the specific very diverse environments, these populations that are indigenous from Peru have adapted and their specific genetic makeup as a consequence of their selection throughout millennia to their specific environments. So I want to start with Caral. So Caral is actually it's an ancient city. Is the oldest ruins of any civilization that has been found so far in the whole of the Americas, both North America and South America. So the oldest civilization that we know of from the Western Hemisphere is precisely in Peru, and is called the Sacred Ancient City of Caral. And I've been lucky to be able to work with archaeologists, local archaeologists, who have now collected 30 samples from coprolites. So coprolites are excrements from humans that have become rock, and we can analyze those samples and look for the microbiome, for the microbes that lived in there, which have also been petrified. And so I've had the privilege to be able to analyze these samples and then compare them to the microbiome of a location called Huánuco, which is not far from Caral. And So we were able to compare samples from current populations from a nearby city to the old Caral coprolite populations. We compare the 6 samples from sewage water in Huánuco versus 6 samples from coprolite, 30 samples of coprolites from Caral. And then the question that we had in mind is, to what extent that civilization that didn't know anything about antibiotics had changed the genes that provide resistance to antibiotics compared to the new populations. In other words, we are looking at antibiotic resistance inherited from those 5,000 years ago inhabitants versus people who are living today. We look at the resistance genes for the main antibiotics and compare the amount of resistance the difference in the amount of genetic resistance that has been that can be measured between the two populations, and we basically found a tremendous amount of new inherited resistance to antibiotics. Yes, we see that the ancient samples cluster distinctly from modern populations, and this transformation tells us about diet, about environment, and as I said, about the antibiotic exposure across millennia. I've also had the opportunity to analyze 30 Peruvian populations. Here we have 7 populations: the Matses, Uros, Chopcas, Moches, Trujillo, Cusco, and Iquitos, in which we sequenced, had 150 whole genomes, around somewhere between 10 to 30 for each of these populations. And here we are counting the amount of SNP, single nucleotide polymorphism, diversity that exists in each of these populations. And what this is telling us is that the populations which are most indigenous, so the ones that have had less exposure to interbreeding with Europeans and with Africans, with all of the different mixture that is a melting pot now in Peru, they tend to have less variation in terms of their genetics compared to the ones that are more what we call admixed. Okay? And so if we look at how that relates to other type of data sets, it's the difference is staggering. Okay, so I have presented to you data from 6 different publications in what you've seen in this presentation so far, and there are 2 common threads that I think involve all of these research points. And there is health equity, the fact that we have this partial representation of disease data because of the invisibility of populations such as those coming from Peru, but also enable harnessing the power of AI. And my main issue, as I will say again and again, is that equity must be the foundation for agentic genomics. You can't build fair future precision medicine built on AI on unfair data. Every tool we create, every agent we deploy must account for who's represented and who's not represented. Okay? So now let's look at the other side of the coin. What can go wrong in the agentic era in genomics? So I'm going to give you things that go wrong independently of whether you are doing genomics or something else, and some things that can go wrong, which are specific to genomics. Okay, the thing that this first risk, which is hallucination and ghost co-authors, which is how I've sort of defined this problem, is general. So it's not just specific to genomics or even science. It's completely general. Okay, we have the subtle threat of numbers that sound really good, that are produced as a consequence of using artificial intelligence, but they aren't. Method sections describing unperformed analysis, statistical results that are plausible but fabricated, and how convincing what we are given from large language models is, because it's all trained and based on things that are probable and most likely. And coming back to these underserved diseases, if you have malaria or schistosomiasis or Ebola, for which there's very little information, if this information that is very little, the large language models have not been fed with that information. So when there is lack of information, the large language model is going to make it up to make it sound as if there was, because it's going to predict based on what it knows, and it's going to infer even if it doesn't have any evidence, because that's precisely what they've been trained for. Okay, and we shouldn't see this as a bug, but more as a feature, right? And also the other thing is that if you are trying to write any essay or any kind of research using large language models, it's going to fabricate references like there's no tomorrow. Okay, LLMs hallucinate author names in citations, fabricate references with plausible digital object identifiers, citations that result to nothing. So every single number, statistic, citation that you have within some corpus of knowledge or text that has been synthetically, that has been generated using generative AI, you need to check it. And ultimately, you are the responsible person who submits that particular work. You are ultimately responsible for that piece of work and making sure that everything is verified. So although artificial intelligence will give you value, you never know what it is that is making up, and you never know what's actually real. You always have to check everything. You cannot take it at face value, otherwise things will go tremendously wrong. And I think that's precisely where the new skills for the future are going to be, and where precisely the bottleneck is going to be, in the verification, the validation, in the making sure that whatever we produce is actually verified and certified. Don't fear intelligence. What you need to fear is invisibility. The real risk isn't being replaced by AI. It's being left behind while others harness it effectively. So the gap between augmented AI researchers and traditional ones is going to only dramatically increase. I'm not going to say in the next couple of years. I'm going to say in the next few months. So I would really invite you to try to get into this new wave of artificial intelligence, not necessarily agentic genomics. As agentic genomics is perhaps a little bit too advanced, but if I were you, I would make sure that not just become an user of this, but you actually become a builder, that you start experimenting, tinkering, testing new models, and incorporate it to the day-to-day activities. Because the sooner you have that AI fluency, the more advantageous your capacity to survive this new wave is going to be. And I don't believe that there are going to be fewer jobs. What I do think is that many of current jobs are going to be destroyed and many new types of jobs will be generated. But for that, you need to harness the power of AI. This idea that I mentioned earlier with the semantic similarity index, that we're going to have bias amplification, where the different biases that the current scientific enterprise depends on actually compounds. And the gaps with the augmentation as given by artificial intelligence increases the differences and the disparities exponentially, right? Unless we engineer by design the models for health equity, the disparities are only going to increment exponentially. That's what I think. We're going to have model bias that becomes agent biases, that becomes systemic biases, and yet genomic data is uniquely sensitive, and so safety and provenance tracking are going to be essential. Okay. So now that I've put a little bit of fear in you, let's be a little bit more positive, and let's as we say in Spain, let's grab the bull by the horns. Okay, there's no other way. We can't escape it, so we better just face it. So how are we going to survive, and how are we not going to drone this agentic in this agentic genomics new era? So coming back, cross-validate everything. Know your models. You know that, for instance, ChatGPT is better at fluency. Claude is better for analysis and figures. Perplexity is better for citations. Gemini is better for, or best for, integration with anything to do with Google tools. And generally, you know, Nano Banana, for instance, is incredibly good for images. So, and then if you don't pay for any of these models, I'm not saying that you should do it. I'm telling you just my own personal experience. The models that you can get for free are substantially less advanced than the ones that you get by paying. I'm not saying that you should pay all of them. I can say that I'm currently paying for 5 or 6 different models, and I use them actually as a swarm in a way, swarm, agentic swarm. So and I call this the dragonfly principle. You may have heard of dragonflies. In fact, dragonflies, they are the most tremendous predator that exists, the most efficacious predator. I mean, if you had dragonflies the size of humans, we all would be extinct millennia, you know, millions of years ago. They are so terrible and specific. And because they have 10,000 lenses in their eyes. The dragonflies have 10,000 lenses. So in other words, their capacity for vision is incredibly well recognized, because they can see from 10 different perspectives, 10,000 different perspectives. And that's why they are so precise. So the dragonfly principle is where you actually use each of the different agents, each of the different models that you have as a different type of impersonation. So let's say that I am writing an essay, yeah? And so you're going to have one agent or one model or one chat that is going to impersonate your tutor, another that is going to impersonate your mother, another one that is going to impersonate your politician, I mean, this is a little bit silly, but you can see that these different impersonations, when you then ask them to evaluate the same model, is going to, or the same essay, is going to give you different perspectives. And those different perspectives can be incredibly valuable when you put them together, because the sort of strengths from different models will be summed up, and the weaknesses will be compensated by each other. So this is a sort of dragonfly approach where you actually evaluate your output, your writing, your essay from different models with different features and different perspectives, in a way that will improve significantly what you end up producing. That's what I call the dragonfly principle. And again, some tools are better for literature review, some others are better for summarization, some others are better for code creation. So make sure that you cross-validate everything. Always benchmark against gold standards, and never trust agent output without ground truth comparisons, okay? Never send anything without checking, yeah? Especially if you are dealing with clinical results, every report must pass through human review. Value your typos and verify your references. Every model every model may fabricate citations. Make sure again that everything that you submit has been validated by a pair of eyes. So intelligence becomes cheap. What's now expensive is eyeballs. Okay, so eyeballs is the expensive commodity now. And remember that you're still biological, right? Yeah, that everyone, not you alone, everyone is experiencing FOMO, fear of missing out. There's no way anyone in the world can keep up with everything that is being released, made accessible at a faster pace on anything to do with AI. And so what the strategy that keeps me going, and that in the end I think has given me a little bit of an edge, is the fact that you have to trust more than ever your own personal instinct, and do not be intimidated by all of the flashy stuff that you see next Twitter or on LinkedIn. Try to harness AI in boring ways that are useful to you. Things like a little application whereby it helps you find literature relevant to your interest. That's not as flashy as the app that I showed you with my robot, but I was there before, and the people who are now so flashy, doing this flashy thing, is because they've been there for months. So make sure that rather than being a passive consumer, that you actually build things and test things that are specifically useful to you. And the obvious things that will be useful for you is to identify the energy drains. Okay, for me, energy drains could be marking, or it could be, let's say, I don't know, uploading something into Blackboard. Or so are there any ways in which you could automate those things that are pain points for you, that will be for your own perspective useful? Okay, that's really what I mean by it. Don't be overwhelmed. Just like swimming, just plunge yourself into the swimming pool and try, tinker, and adopt and experiment. That is really what's going to give you the edge for what's coming. So what's the future going to look like? So one perspective could be the tool that I'm currently developing, which is called ClawBio, which is the agentic biology stack. So I'm going to introduce now ClawBio. So as I said, the way artificial intelligence is going to work is mostly through agents. And as I said, agents are programs softwares that run on their own, that are autonomous, and they have intelligence. And you can communicate with them and ask them things on plain English. So ClawBio is the new repository that collects as a central registry biological skills, like the one that you saw, the drug photo. Yeah, that's one skill that that can be pushed, shared, reproduced, and reused by someone else. Okay. If you imagine the way currently biological knowledge is being spread, it's mostly through PDFs, through articles. If you try to reuse any of that functionality into, you see a paper you're interested in and you want to reproduce how they did it, you have to go and retrieve the data, retrieve the code, and so on. And of course, that which is difficult for a human would be even more difficult for an agent. So ClawBio is a repository that is able to package those skills, code, data, instructions, all into a very, very easy command, just one command that you need to type on your computer, and you get all of that functionality in a reproducible, self-contained manner. And so the same as you will have a library where you have all of those knowledge in books contained and discrete, you can take that particular item in the library and use it. And you're going to have that specific functionality working undiscoverable by your own agents. Okay. So why is this important? Because domain-specific agents can transform genomics faster than general artificial intelligence. And the good thing about this, specifically in the biomedical health data, is that your own data, health data, is incredibly sensitive. And ClawBio allows you to get the skill and apply into your own personal vault of data without having to share it. Okay, so that makes it into a very desirable type of functionality that you would want to work with. So if you're interested, ClawBio is accessible on the internet. If you go to clawbio.ai, you'll see the project website. And if this is something that you're interested in, you can actually reuse it or ask me questions. So here you can see this is ClawBio. So ClawBio was started 14 days ago, approximately. And if you click on here, it has been downloaded more than 2,000 times. And people using this repository of code centralized called GitHub, I've had a huge amount of people who have expressed interest. You can see again the same functionality I demonstrated with warfarin. That's a particular skill of ClawBio. So all of the stuff that I showed you before for my robots is actually functionality that exists in ClawBio. So feel free to have a look if this is something that interests you. And as I said, we have tremendous amount of social proof that has already shown significant interest in this development. So let's now look at the final slides. So if there are a number of takeaways in terms of what I like you to go home with are these. So first of all, deep intelligence is the bottleneck, not the processing power. Those people that think that your brain is going to be atrophied, I think you are in for a nice surprise, because that sort of gut instinct decision making that humans have is something that I feel that is now developing more than ever. And if you think that artificial intelligence is going to take away the need for you to having to learn new things, I think you are probably also waiting for a nice surprise, because there another approach can be those people who actually use AI to empower them in terms of understanding. And me, who is someone who is currently using around 10 different AI agents any time of the day or night, I've learned more than ever before in this particular year, because the pace of development has significantly increased, and as a consequence, there's a lot more that I need to learn more faster. So for me, I can see why other people would feel that your brain can atrophy, but I think there's also the other side of the coin, where you're going to have people who use this to even empower them more in terms of their cognitive ability. Because one of the things that AI allows you to do is to be able to focus your mental cognitive load into those things which truly require the strong thinking and the deep thinking. And that's where I think that AI can have the greatest impact possible. Coming back to the key takeaways, I also believe that equity must be built in from the start if we are going to solve or bridge some of the current health disparities that exist. Otherwise, I expect these disparities to increase exponentially. Make sure that you verify everything: figures, numbers, references, author lists. You can't take on face value anything that an LLM regurgitates, because it's going to produce things that sound really, really nice, but actually are totally and completely fabricated. I think that the real key is to make sure that you are incredibly expert in one particular type of area, where you would be able to know more nuance than what the global generalist large language models represent. So all of the models that we use, Claude, ChatGPT, they're all generalist models, and they're not going to perform so well on things which are niche, which are really deep. Okay, and that's where I think the opportunity for those of us who want to survive the coming wave can be, right? And also, as you can see, I have my own research lab, and I have around 10 AI automated helpers. And I think this is something that's going to be, it's already the reality for some of us, research leaders, people who work in the academic world, but I think this is going to be the norm. Everyone who is a knowledge worker is going to have a set of agents, and these agents are going to be specialized on your specific context, making decisions for you that are trained with your own way of thinking throughout the years, and that's what going to give you the edge in terms of more productivity, in terms of more focus and decision making. All right. So with that, I'd like to thank you for your time and for listening. I hope that this was a little bit enlightening. Certainly, that's the thing that I want to leave you with. People think about super intelligence as the goal of AI. I think that's exclusive, and I don't like it. Other people think about super abundance, AI, the future is going to give super abundance. I think also that's selfish. I think the purest purpose I found in terms of what helps me embrace is if I'm going to use AI for my own enlightenment, for achieving greater wisdom, and for making better decisions. So thanks very much for listening, and I look forward to any questions that you may have.